This week, you're going to make your first self-assembled monolayers, and we're going to characterize them using your contact angle instruments. The substrates that you'll be using this week will be glass and a silicon TiO. So glass is just silicon dioxide, and it can have two different forms. It can have a regular crystal structure, and in that case it's quartz crystal. And so if you have anything that's quartz, you know that this is regularly ordered. It's very nice and good crystal structure. But regular glass is amorphous, and it does not have a regular crystal structure. So you can see it's kind of random, kind of messed up. There's big pockets. A lot of times there's sodium atoms in here, that sort of thing. So we're not going to be using quartz because, as you can imagine, that's probably a little more expensive. But we have glass slides for sure. And we're going to be representing that surface. Here's the substrate, the silicon dioxide. But at the surface, we have some silicons, right? And we have some oxygens, and sometimes they're bound to each other and sometimes they're not. And so this is what our glass substrate would look like. Silicon is pure silicon, has a tetrahedral structure, a covalently bonded tetrahedral around every single silicon in a regular ordered pattern. And so we would represent that substrate as a block of silicon, right, with silicon atoms at the top in a regular pattern. And then TiO is tetraethyl orthosilicate, okay, which has this structure. It's a silicon with a tetrahedral structure of oxygens around it, and then each oxygen has an ethyl group, which is CH2CH3. So a little bit of an organic linker on the end. Now, when you add TiO to the substrate, it creates a monolayer on the top of the silicon that looks a lot like glass. So you've got silicon dioxide here going on the surface, and so we're going to represent that silicon TiO substrate. We could do it this way. We've got our silicon substrate, which had our regular order of silicon, right? And then we put the TiO on top, so we've got these ethyl groups on top. And that would be our silicon TiO substrate. Now, when we clean the substrate, we clean the glass or the silicon TiO with concentrated sodium hydroxide solution, and that has multiple purposes. So the first purpose is to remove any dust or dirt from the surface. Also to remove any organics, oils, greases, etc. From people touching it, from fingerprints, etc. Or from the manufacturing process. It also has the effect of preparing the surface through hydroxylation, so that all of the termination points on the top of the glass, any silicon is now attached to a hydroxyl group. Same thing goes for the TiO, and all of the ethyls get removed, and you end up with just hydroxyls on the top. So these two substrates are very similar, although glass is transparent and silicon is like a silvery reflective substance. And so the actual bulk of the substance is different, but the surface is very similar. Now the hydroxylated surface on the TiO is probably more regular than the glass because the glass we're using is amorphous. But essentially, we have very similar surfaces here between the glass and the silicon. The SAM molecules that you're going to be working with this week are amino silanes. So when we talked about SAMs in the first week of lab, we told you a little bit about the structure of a self-assembled monolayer molecule, right? There's a head group, and that attaches to the surface, and then there's a functional group which is now on the top, okay, on the other end of the surface. So the head group in this case is a silane or a silicon end, okay, so silane would be ANE, meaning there's some carbons and hydrogens there, but the SIL is silicon, okay. And you can imagine that that's going to bind to the silicon surface, right, or a silicon oxygen bond. Okay, the functional group, or the part that's going to replace the surface, is going to be on the surface now, is the amine, or the amino group, the NH2 end. So here are just two examples of our amino silanes. You can see there's a silicon end with some oxygens and organics, okay? So this looks a lot like the TiOS molecule, right? And then there's an extra piece here, and that is the aminopropyl piece. So propyl means 3, 1, 2, 3 carbons, okay, and then the amine on the end. So this is the functional group, and then this silicon is going to actually bind to the surface, and all of this extra organic stuff is going to be eliminated from the molecule. Same thing here, 3-aminopropyl trimethoxysilane. So here we just have less organic attached to the silicon, whereas here we had an ethoxysilane, so we had more organic here attached to the silane. When you soak your slides or your substrates into a SAM solution, so for example, aminopropyl triethoxysilane, okay, your silicon is going to be the thing that binds to the surface, the oxygens here, okay? So the oxygen and the ethyl part, the carbon, CH2, CH3, right, that whole end is going to be eliminated, right? It's going to pull this hydrogen off and create ethanol, and that's going to go into solution. But the silicon then is going to bind itself to the oxygen that's left. So if this can have multiple binding sites, it can bind multiple times to the surface, or it can bind to other SAM molecules that are next to it, those silicons. In either instance, we're going to create this layer of silicon oxide here on top of our surface, and then everything that's pointing up away from the substrate is going to have the aminos on the end. Now, there will be intermolecular forces in between each one of these amine groups, in between each one of these linker chains, okay? And so hopefully we get a nice ordered structure here. So it's kind of like a crystalline thin film on top of our glass. But we've essentially replaced a hydroxylated surface with an amino surface. We can represent that in a different way. Here's our slide or our substrate that's a glass, and we're hoping that all of the head groups, all the silanes, are going to covalently bind to the surface, and all of their tails and functional groups are going to be pointing upwards and replace the surface essentially. Now, how do we do this? A general procedure, a good starting place, is that we're going to take our clean glass or TiOS slides, we're going to soak them in a SAM solution. 1 millimolar is a good reference point that you can soak in solution for 30 minutes. Once your 30 minutes is up, you can take your slides out of the solution. You can rinse them with deionized water, so take a little squirt bottle and rinse them off, and then gently blow them dry with the bench air. So that should push all of the water off the edge so that you have a dry surface. Then you're going to measure the contact angle of a clean side and measure the contact angle of a self-assembled monolayer slide, and you're going to try to compare which is more hydrophilic, which is more hydrophobic, what do you expect to happen, okay? And you're going to use your contact angle apparatus that you optimized last week. You will have the opportunity to ask your own questions and create your own expectations for this lab.